All right, let's get started. So there have been a number of questions about the Connect website. So when you go into the Blackboard website, this is approximately what it should look like for you. On the left-hand side, if you click on Units and Lectures, Unit 1 is the only one that's up there right now. If you click on that, if you click on Unit 1, you should have a table of contents. So Lecture 1, if you click on that, it has the PowerPoint for Lecture 1. If you click on Lecture 2, it has the PowerPoint for Lecture 2. If you click on Chapter 2, notice how it says Learn Smart. This is the link to be able to get to the reading assignment for Chapter 2 on the Learn Smart website. So if you click on that, it should automatically take you to their website, and that's where you would enter your username and password information in order to get in. If it's the lab, so lab safety is the first lab you have due. It says Learn Smart Labs. Gives the due date. Again, you just click launch and it should take you to their website and you sign in order to do that assignment. Does anyone have any questions? All right, again, I do record the audio for each lecture, and I post that as well. So after each lecture, you'll see Lecture 1 audio or Lecture 2 audio. If you click on that, it should open up a new page, and you can listen to me until you fall asleep. I've actually offered it as a way of treating insomniacs. It's awesome. Okay, we left off right around here talking about Cox postulates. Did we get through Cox postulates? No? Okay. We talked about Edward Jenner. I thought that's... Malaria. There we go, right there. Okay. So some microorganisms are good. We use them for making cheese and all sorts of cool things. And some microorganisms are really bad, like malaria. So somewhere between 500,000 and 750,000 people die every year from malaria. Generally, this is in the tropical countries in the world where the mosquitoes that carry malaria are found. So disease can truly shape the world. Smallpox has wiped out entire civilizations. In 1918, there was an influenza pandemic. It killed 50 million people in 18 months. 1918 was also the year that World War I ended. 11 million people died during World War I. It lasted 4 years. So as human beings, it took us 4 years to kill 11 million people. And in 18 months, one virus killed 50 million people. So it makes human war look stupid. Like this virus does way more damage than we can even do to ourselves. So truly, history is shaped by infectious disease. All right, we have this kind of golden age of microbiology where a lot of the obvious diseases were found. Things like tuberculosis and measles and polio, the things that we commonly think of when we think of infectious disease. So basically the easy stuff is done. Now, as microbiologists, we're left with the more complicated thing. So today we now know that female infertility is caused by chlamydia infection. So if you get infected with chlamydia trachomatis, it damages the fallopian tubes, and then you can't have children. But we didn't realize that that's why infertility happened before. We have things like liver cancer, which is caused by hepatitis virus infection, or cervical cancer, human papillomavirus, ulcers caused by Helicobacter pylori. Coxsackie virus is now associated with diabetes and schizophrenia. So mental disorders may actually be associated with an infectious disease. All right. And we don't even do a web lecture on this. Just raise your hand. All microbes cause disease. Who thinks that's true? Who thinks it's false? Good. Okay. So Robert Hooke was one of the first people to observe microorganisms. He made a very crude microscope, and he was able to see some of the larger fungi. He looked under a microscope and he saw these spore-forming parts of fungi. A little bit later, Anton van Leeuwenhoek made a little better microscope, and he was actually able to see probably some larger bacteria. He called them animalcules, or little animals. He got it from his teeth, too. He scraped plaque off of his teeth, and that's where he found it. All right. Louis Pasteur, great microbiologist. Not only did he discover a lot of things about microbes, but he also disproved spontaneous generation. So before his time, people thought that meat rotted or flies came from just, I don't know, they would just spontaneously appear. What he showed is that there is no such thing as spontaneous generation. Life begets life. If you want bacteria to grow, you have to have bacteria present. They don't just pop up. And the way he did that is he took this weird-looking flask and he boiled it, killed everything in there, and then he just let the flask sit there. Or he took another flask and boiled it, and then he broke off the funny little neck on here so that some of the germs floating around in the air could get down in here. So this flask started growing bacteria, and this flask never grew bacteria because the bacteria can't go all the way up this tube. And in fact, this flask is still sitting in a museum almost 200 years later, and there's nothing growing in it. So there is no such thing as spontaneous generation. All right, Edward Jenner. So he is the first person to really develop a vaccine. So he developed a vaccine against smallpox. He found that if you vaccinated people with cowpox, cowpox is similar enough to smallpox that it actually kept the person from ever getting smallpox. He went about it in a little unorthodox way. We don't like to do human experimentation, and he did. He just randomly picked people and started sticking them with cowpox to see what would happen. That's really frowned upon nowadays, but he proved his point. Ferdinand Cohen, he discovered endospores. So he found that certain bacteria make spores which are very resistant to boiling chemicals like bleach. So if you work in a hospital, there's this really nasty bacteria called Clostridioides difficile, or C. diff, and it's a pain in the butt, literally. It's really hard to get rid of it. You can bleach a room, you can do all sorts of things, and the spores are so resilient that it just keeps hanging around. All right, Louis Pasteur also invented pasteurization. So when you pasteurize your milk, you can thank Louis Pasteur. He showed that human diseases could arise from infection. So he showed that you could take a bacteria and make someone sick with it. A lot of this stuff seems logical, like, yeah, duh, but no one knew it back then. And then Robert Koch. Koch came up with a list of experiments that you needed to do in order to prove that something was a pathogen. So he said, and we call these Koch's postulates, he said that the microorganism must be present in every case of the disease but absent in healthy people. So if you've got the flu, then we should be able to isolate the flu virus from you, but not the person next to you who doesn't have the flu. If we look at Staph aureus, it grows on the skin of most people. Doesn't usually cause a problem. The suspected microorganism must be isolated and grown in a pure culture. So you have to be able to purify it, and then you have to be able to take that pure microorganism and re-inject it, in this case into a guinea pig, and show that it causes the same disease. So you have to be able to purify that pathogen and cause the same disease in something else. He was using tuberculosis when he was doing his experiments. And then after the disease, so once our poor little guinea pig dies, you should once again be able to find that tuberculosis bacteria in the animal. So this is good, and in most instances we are able to do this. The problem is some microorganisms we can't culture. So what's syphilis? Come on, you guys know what syphilis is. Really? What was that?
The reason we left Vietnam.
I suppose that's one reason.
It's
a sexually transmitted disease. Thanks. So it is a sexually transmitted disease, but you can't grow it in a petri dish. And so we actually can't fulfill Koch's postulates because we can't grow it. But that doesn't mean syphilis doesn't cause a sexually transmitted disease. It does. So nowadays, we have more sophisticated methods, DNA sequencing, things like that we can do to prove that microorganisms are causing disease, even if we can't grow it and isolate it. All right, Joseph Lister. What's that sound like? Listerine. So he is the first person to utilize hand washing and misting in operation rooms. So he had this cool little device, and he would spray an antimicrobial substance, in this case phenol, on the operating surface to sterilize it. And not surprisingly, his patients did a whole lot better than most patients. Oliver Wendell Holmes and Ignaz Semmelweis, they showed that women became infected in the maternity ward after physicians had been working in the autopsy room. They weren't washing their hands, so they were dissecting someone who died from some weird infection, and they would go straight into the delivery room and deliver a baby. Not a good idea. Unfortunately, Semmelweis basically went crazy because he talked about this. He told all of the other physicians he was working with, hey, we need to be washing our hands. And they were basically using a bleach solution to wash their hands. If you've ever washed your hands in bleach, it's not very friendly. It starts hurting your hands after a while. And so all the other doctors were like, we're not doing this anymore. This is stupid. And so they went back to doing what they were and killing babies. And so he went crazy as a result of it. But he was right. All right. Okay. So spontaneous generation was disproven by which of the following scientists? All right, so Louis Pasteur did indeed disprove spontaneous generation with his swan neck flask. All right, last couple of slides for lecture 1, and then we'll move on to lecture 2. If you want to be able to do well on your lecture and lab exams, you have to know how to properly write the name of bacteria or other microorganisms. So, genus and species is always the way that we refer to an organism. So, Homo sapien, human, the genus and species. Pseudomonas aeruginosa. Pseudomonas is the genus, aeruginosa is the species. If you're typing on a computer, you can italicize it. If you're writing it by hand, I can't tell if you're italicizing it. So you have to use underlining. So you write genus, and it's always, first letter is capitalized in the genus, and then the species, no capital letters, and underline it. And you may see it abbreviated sometimes, E period coli. Even though that is acceptable, it's not acceptable for your exams. And the reason for that is I don't know that you know what E means. I need to know that you know that E means Escherichia, not Enterobacter or something else. So you have to write out the entire genus and the entire species in order to get full credit on your exams. All right, so that's for bacteria. Viruses are different. You don't capitalize and you don't italicize or underline a virus name. So if it's pox virus, you just write pox virus. You don't have to capitalize it or underline it or anything. Okay. You may also find instances where there is a serovar or a strain. This is the same thing as a breed of dog. So dogs are all the same species, but a greyhound and a chihuahua are different breeds of dog. Well, you can have a strain or a serovar of bacteria. So E. coli DH5 alpha or E. coli 0157H7. They're both the same species, they're just a different strain, like a different breed of dog. And it makes a difference. DH5 alpha is used, that's what we use in lab. It's basically harmless. 0157H7 causes the really nasty diarrhea outbreaks from McDonald's or wherever you get food contamination. So, just like a Chihuahua and a Greyhound don't look the same, the effects of these E. coli on your body can be completely different. Even though it's the same species, it's just a different strain. All right, there's also a pronunciation guide in Lecture 1. I'm not going to go through all of this. It is helpful to know how to correctly pronounce things. If you walk into lab and you say, where's the tube of... your lab instructor is going to look at you like you came from Mars. So take some time and at least come close to the proper pronunciation of the bacteria. One of the easiest things I found is just type it in on Google. Type in, how do you say Escherichia coli or Pseudomonas aeruginosa, and it will usually pull up a link to a website where it'll say it for you. So, all right, another clicker question. What is the proper way to write that on an exam? Okay, good. So A is the correct way. Make sure it's underlined. All right, let's go to lecture 2. So microscopes, tools, and classification. So by the end of this lecture, these are the things that you should know. You should know the different parts of microscope and how a microscope works. You covered that in lab some. Know the approximate size of different microbes. So I don't expect you to memorize the exact size of every microorganism, but you should be able to know if something's bigger or smaller. So if I say, is T4 bacteriophage virus bigger or smaller than E. coli, you should be able to tell me which one's bigger or smaller. Be able to compare and contrast the categories of stains and provide examples. The 5 I's. So this is kind of a scientific method, how you go about examining a bacteria. You should know by the end of this lecture the overall differences between bacteria, archaea, and eukaryotic organisms, and the different classes of biological molecules. So we're just briefly going to go over a little bit of the chemistry here. Again, I expect that you know what a covalent bond or an ionic bond is. So if you don't remember those things, you're going to need to go and read through those sections of Chapter 2 and make sure that you know what they are. All right, prokaryote versus eukaryote. In general, prokaryotes are about 10 times smaller than eukaryotes, and prokaryotes don't have any organelles. So here we have a bacteria or an archaea. It's got a cell membrane, it's got a cell wall, but there's no nucleus, there's no mitochondria, there's no chloroplasts or anything inside. In the case of a eukaryote, they in general are bigger. It's not always true, but in general they are bigger. Viruses, we'll cover in the next unit. They're not alive. It's just a piece of DNA with some protein covering it. And they tend to be a whole lot smaller. All right, in the study of microorganisms, we have helminths, so these are worms. So even though an infection with a parasitic worm, the worm is big, you can see it without a microscope, we still consider it microbiology because the eggs, which is what you actually infect yourself with, are microscopic. And so the transmission of the disease is still of a microscopic size. We have fungi, we have protozoa, we have bacteria, and we have viruses. All right, the majority of all microorganisms on the face of the planet live just in the soil and the water. Most of them are harmless. They don't cause any disease. In fact, most of them we can't even grow. So we know very little about them. We know what their DNA sequence is, but we don't really know what they do. Parasites, however, they require the host to provide them with nutrients. And all parasites cause damage or disease. All right, so what's the main difference between a bacteria and a fungi? couple more seconds. Okay. So bacteria are, in general, smaller than fungi. Although the bacteria Thiomargarita namibiensis is big enough that you don't even have to have a microscope. So remember, most of the things we talk about are generalizations. Fungi have a nucleus, fungi have organelles, so all of the above is the correct answer. Alright, if we look at life on the planet Earth, we can break it up into the domain bacteria, the domain archaea, and the domain eukarya. If we follow that back to what we consider the last universal common ancestor, some cell billions of years ago, archaea are actually more similar to eukaryotes than they are to bacteria. So we didn't used to think that. We used to think that bacteria and archaea were fairly similar to each other, and then eukaryotes were kind of way over on the side. But when we actually start looking at the enzymes and the way that archaea function, they're actually more similar to eukaryotic cells than they are to bacteria. All right. You should all know the general classification scheme: domain, kingdom, phylum, or division, class, order, family, genus, species. This is our way as human beings of trying to make sense of the world around us. It doesn't always work, though. It's not like when the bacteria were evolving, they were like, all right, now you have to be in this kingdom and you have to be in this domain. They just evolved. So we use it to try and understand the world around us, but there are exceptions. It used to be that we based taxonomy on physical appearance and the behavior of organisms. So we would look at E. coli and we would say, okay, E. coli can use sugar as a carbon source. It's about this big. It's this size and shape, so we're going to stick it over here with these other organisms when we classify it. Now we use genetic evidence. We sequence the genome, and we see how similar the genomes are from one to the other. And as a result of that, sometimes we totally rearrange the classification scheme. In fact, I mentioned Clostridioides difficile, C. diff. Its name just got changed. It used to be Clostridium difficile, and they changed it to Clostridioides difficile because they found out its genetics is actually fairly different than the other Clostridium species. All right, the size of things. In general, you can see down to about 0.1 millimeter, maybe a little bit more than 0.1 millimeter. Anything less than 0.1 millimeter, which is 100 micrometers, down to about 1 micrometer, you have to have a light microscope in order to see that. Once you get smaller than a micrometer, you really need an electron microscope to be able to see those things. So all of your viruses actually fall into the category of needing an electron microscope to be able to actually see them. Most bacteria fall into this range where we can see them with a light microscope. When we talk about magnification, we're talking about making things larger. We frequently refer to it as X, so you would say 10x magnification or 10 times its normal size. I'm not going to go over the specific parts of the microscope because you went over those in general in lab. I guarantee there'll be a picture like this on your first lab exam, and you're going to have to label all the different parts of the microscope. So make sure you take some time to look at the picture of the microscope in your lab manual and learn the different parts. Not only that, when you're talking to someone else, it's a whole lot easier to use the correct term like, oh, my ocular or my eyepiece is dirty, instead of, well, the thing you look through, it looks weird. So that doesn't tell me a whole lot. So if you use the right name, the ocular or the eyepiece, then I know what you're talking about. All right, in general, there are two lenses inside the microscope. You have your ocular lens and your objective lens. And so it's the combination of those two lenses that gets you your overall magnification. So you just take the magnification of the objective lens and you multiply it by the magnification of the ocular lens. So in this case, if you have a 4x objective and a 10x ocular, your total is 40x. Pretty simple math. This is what bacteria would look like at 40x. If you go all the way up to your 100x objective, you're now at 1000x, and this is what the same specimen would look like. All right, there are some other important considerations with microscopes. Just because you make something bigger doesn't mean you can actually see fine detail. So this is like on your computer. If you've ever taken a picture and just stretched it, it becomes blurry. Even though you've made it bigger, it gets so blurry that it's useless. Your microscope is no different. These are the same magnification, but the resolving power of this is much less than this. Here you can see really fine detail. Here it's just blurry. And so the resolving power is also very important. Not just the magnification, but the resolving power. And what resolving power really is the ability to tell that two objects are really two objects. So if you have low resolving power, the two objects actually look like one object. And so to your eye, it then becomes blurry, and it just doesn't look clear. If you have a good resolving power, you can actually tell that these are two separate objects. So what does that mean? If you're looking at a cell and you've got low resolving power, you can see the big stuff like the nucleus of this cell, but you can't see all of the little organelles because you lack the resolving power. Even though you've made it look huge, you can't see the detail. All right, the oil immersion lens on your microscope. So you put a drop of oil on there, you have it set to 100x. You all did this in lab yesterday. The oil immersion can resolve objects as small as 0.2 micrometers if it's a good microscope. So why do you have to have the oil? The oil actually is a special kind of oil, and it causes the light waves to bend in toward the lens. If you don't have the oil, then when the light passes through your microscope slide, it actually bends out away from the lens and it gets lost. And so that lost light means that you have less resolving power and your image looks blurry. So by forcing those light rays to curve in toward the lens, you actually get better resolution. And this isn't just any oil. You can't just take like cooking oil or something and put it on there. That doesn't work. The way this works is the light bending properties of the oil are the same as the glass. And so that way, when the light comes from the glass into the oil, it just keeps right on going. Air does not have the same properties, and so you can see that some of it gets deflected down, some of it gets deflected out. So it is a special kind of oil. Don't just grab any kind of oil. All right. Even if you're using a light microscope, there are different kinds of light microscopes. You can have a bright field, which is what we use in lab, mainly because they're not too terribly expensive. You can spend hundreds of thousands of dollars on an expensive light microscope. We don't have enough money to buy $96,000, $100,000 microscopes. You can also have a dark field. You can see the background's dark. It makes your specimen stand out really nice. Or a phase contrast. You can also have fluorescent microscopes where you can have a fluorescent dye on your sample and be able to see specific structures based on what it is dyed with. A confocal microscope is just a really fancy fluorescent microscope. And finally, an electron microscope, where you can actually start to see cell membranes and very fine structures. Notice the power, 100,000x versus maybe 400x. All right, some different preparations. Wet mounts or hanging drops. So the nice thing about using a wet mount, the organism is in the liquid that it was growing in. So you can study the organism in basically its natural environment. You can see if the cells are swimming. You can see the natural shape and size of that organism. The problem is it can dry out. So evaporation will happen and it will dry out over time. You can also end up contaminating things if you get the drop of liquid on the microscope lens or on your hands. So there are some pros and cons. You can also fix your sample. Heat fixing is what you will be using in lab. So you'll put a drop of your sample on a microscope slide and then you'll heat it. All the liquid evaporates and it causes the sample to stick to the glass. So that's great. It's stuck there. It's easier to stain and do things with. The problem is it kills the cells. So now if you wanted to see if the cell can swim, well, the cell's dead, so you can't see if it can swim anymore. So again, there's pros and cons to everything. Staining. The purpose of staining is to be able to give contrast, to be able to see certain parts of the organism that you want to study. In general, there are two types of stains. You have a basic stain, which is cationic or positively charged, and you have an acidic or anionic stain, which is negatively charged. What charge is the cell membrane? The phosphate groups on the phospholipid bilayer, what charge are they? They're negatively charged. So if you have a positive stain, it is attracted to the negatively charged components. Remember, opposites attract. If you have a negative stain, it has a negative charge and it is repelled by the cell wall and the cell membrane. So what does this look like? If you have a positive stain, the bacteria are now stained and the background is white. If you have a negative stain, the bacteria are not stained, so they are white, and the whole background is now stained. We have simple stains, which means we just want to stain the bacteria so we can see it. A lot of times bacteria are very clear and they're hard to see under the microscope. So if you can stain it with something purple or violet colored, now you can see the bacteria much more clearly. You can also have a differential stain. So the Gram stain is one that you will be doing in a week or so. And it allows you to differentiate between different types of bacteria based on their cell wall. So we call it a differential stain because you can use it to differentiate. There are also other types of stains for endospores or other things that allow you to differentiate between different types of bacteria. The capsule stain, some bacteria have a sugar coating or a capsule around them. The bacteria is actually the dark spots in the middle. The clear area is the capsule, and then the background, which is just slide. All right, we're actually going to skip this question because of time. All right, the 5 I's. So inoculation, incubation, isolation, inspection, and identification. So this is what you do anytime that you are studying a microorganism. You're going to inoculate it. You're going to take a sample and grow it on a plate or a broth. That is inoculation. You are going to incubate it. Could be in the incubator, could be on the desktop, depending on the temperature it grows at. Isolation if you took a throat swab, there's going to be tons of kinds of bacteria at the back of someone's throat. So you need to be able to isolate the one you are interested in out of all of the other ones. And then inspection and identification. So inspection could be looking at it under a microscope, staining it, examining what type of sugar molecules it can live off of. And finally, you would use that information to identify your microorganism. So sample collection, it could be a cut, it could be a blood sample, it could be a urine sample, many different types of samples. You can then put it onto a plate. You could put it into, maybe it has to grow in blood. Sometimes we inject it into chicken eggs. So flu virus grows in chicken eggs. That's how we grow flu virus. Finally, you incubate it. And then you may come back with a ton of different kind of bacteria, and you have to isolate the one that you want in order to study it further. As I said, you may run biochemical tests, immunological tests. You might have an assay to see whether a person has antibodies against a particular pathogen, or DNA analysis. All right, in this whole process, sterile technique is absolutely essential. If you cough on the sample, well, you don't know what came from the patient and what came from you. So it doesn't matter where your sample came from, whether it's a broth in the lab or a sample that a doctor just sent to you as a clinical laboratory pathologist. You need to have exceptional sterile technique. We have what is known as a mixed culture. So that is a sample that is intentionally has more than one microorganism. So again, if you take the throat swab, it has more than one microorganism in it because that's what it has. It just came that way. A contaminated culture started off as one microorganism, and because of improper handling, you contaminated it with something else. So mixed culture is fine. Contaminated culture is always a bad thing. You can look at this. You have two different organisms growing on this plate. One of them's white, one of them's yellow. That's just what came from your skin. There's multiple organisms growing on your skin. So this is normal. You may want to then separate them out to study them further. A contaminated culture started off as one thing, but now you can see this white contamination in there because you weren't using proper sterile technique. If this is a patient sample, and you look at this and you say, oh, well, maybe this person's infected with two things, so the doctor prescribed something completely different, when in reality they only needed to worry about the red one, not the white one. Okay, so we'll stop there.